and the recordings will be available in my YouTube channel. So this is the link for that, or you can simply search Mochandra PMRF IIT Kanpur. You will get the YouTube channel, as well as the notes will be shared in this Google Drive. All these related documents will be available or will be made available to you by NPTEL people in the course platform as well. Okay, and during every class, I'll be taking the attendance. So I'll take the attendance at the end of the meeting. So, a few housekeeping rules for this meeting, which you guys have to follow for every meeting, basically. So you guys are joining this meeting as a guest, so which is why you won't be able to see the chat option, but you can always unmute yourself and ask me queries directly. Okay. Otherwise, you can raise your hand. Okay. And I can maybe call upon you and then you can unmute. But in between, if you want to ask a specific query related to a specific slide, you can definitely unmute yourself and ask. But please note, if from your side, if any undisturbed, like some sound or something like that is coming, or you're unnecessarily creating nuisance during the meeting, then I'll have to remove you from the meeting. Okay. So please try to maintain decorum of this meeting so that we can learn more effectively. So let's begin with the content. Let me, so are you guys able to see my screen? I think it has frozen. Am I audible?
Yes.
Yes. So are you guys able to see my screen?
Yes.
Yes,
Um,
ma'am.
okay. Okay. So so this is the slide that I'm showing right now is what is biomaterial. Is it visible to you? Or are you yeah?
Visible, visible.
Okay,
No, ma'am,
okay.
not.
Not visible. So actually I asked this question because I've joined from two devices and one of my device it is not coming. That's why I asked. Oh yeah, now it should be visible to you, all of you. Okay. So maybe some net issue. That's all.
Yes, ma'am.
So let me ask you a very basic question. So the professor who is taking this course, he also explained, right? What is a biomaterial? So we will revise those concepts during this problem solving sessions, as well as we will look into few sample problems so that the concepts become much more stronger in your mind. So the images that you can see in this slide, I want you to guess which of these are biomaterial, or you can call it a biomaterial. So any one of you would like to give it a try. So figure A. Do you think this is a biomaterial? Anyone? Yes, no?
No.
No,
You're
ma'am.
correct. Correct. So this is not a biomaterial. What about figure B? Is it a biomaterial?
No.
No. Okay. So this is actually a biomaterial. You know what this is? This is a hip joint replacement prosthetics. So the surgeons use this. So this is the ball and socket joint that goes at the um, at the here in the hip in the pelvic bone, whereas this part goes into the femur. So this is actually made up of biomaterials like stainless steel, titanium, etc., and used to replace the hip joints. Okay, so this is actually a biomaterial. So what about this figure C? Do you think this is a biomaterial? Or do you know what this is?
No.
So this is basically a vascular stent, okay? And these stents are usually made up of, again, various kind of polymers like diachron, etc., or metals, titanium, stainless, etc. So these stents, they help in cases where the diameter of the blood vessels have become smaller and the surgeon wants to open it up. So they deploy these stents inside the blood vessel and they open up. okay, at that particular location, and then restores proper blood flow. Okay, this is a biomaterial. Then, do you know what this is? This figure D, the last figure. Anyone?
Implant.
Yes, this is a silicone breast implant. Okay, breast implant, and these are made up of silicone. That's why the name silicone breast implant. This is a biomaterial. So from these figures, what we could understand is a biomaterial is usually any substance. Now, this substance can be of synthetic origin, natural, or it can be a combination of both. Okay. So, and these biomaterials, they are used either for some time, like for a very short duration, or for years, or for the entire lifetime of the patient. Okay. And these materials, they are used either self-sufficient. They are either self-sufficient on their own to function, or they're used in combination with the human system, like any organ or tissue, so that together they are able to do some functions, like they can treat or help. Augment means they can help any process or help in the diagnosis, okay, or replace any tissue, organ, or function. So, uh, in general, so any material which are used in basically in the body, okay, and they are of synthetic, natural, or in combination type of origin, they're used for short duration time, or it can be used for very long duration, like throughout the lifetime of the patient. And they're used for some application, like they can be used for the treatment, particular type of treatment, or for diagnosing something, or to replace some damaged tissue, organ. Okay, so this is the definition. Or we can also say that a biomaterial is a non-viable material which are used in medical device, and they are intended to interact with the biological system. Now, if we remove these keywords non-viable and medical device, then the definition of biomaterial becomes much more broader. Okay. So nowadays, the fourth generation biomaterials, like tissue engineering constructs, they are called. So these materials are viable because they contain the living cells. And these materials now again can be classified into the broad umbrella of biomaterial. And there are also those type of materials which are not necessary for a medical device purpose, but used for some other application. Okay, maybe some type of a sensor, okay, external or internal sensor. So again, that this definition of biomaterial is very broad, but the thing that remains constant in all this definition is it has to interact with the biological system and do some biological function. Okay, so that is true for all definitions. So I'm thinking that you guys have understood the definition. Now I'll come to this small question. So this question is, which of the following is or are biomaterial? Okay, so there are four options given, and you have to choose any one of them. So would anyone of you like to try what will be the correct answer here?
First, second, and fourth.
First, second, and fourth. So you're correct. You're correct. The answer is first, second, and fourth. The third option here, this is not a biomaterial, or this type of devices doesn't come under the broad class of biomaterial. Whereas the materials which are used for tooth filling, or for cardiovascular repairs, or for hip replacements, these materials can be classified under biomaterials. Is good. So any one of you have any additional queries to this question, or is it understood to everyone?
It's understood.
Great. Uh, so let's move on to the next question. So which of these, like there are four options, is a biomaterial? Penicillin, orthoscrew, metmorphin, or a thermometer?
May I answer?
Yeah,
Orthoscrew.
yeah, sure. So you're correct. The answer is orthoscrew. Why other threes are not the correct answer? Here, penicillin is a drug. So, drug cannot be classified under biomaterial, as well as the metmorphin is also a type of a drug. Whereas the correct answer, ortho screw, are basically metallic screws which are used by orthopedic surgeons for fixing small fractures, okay, in various kind of bones. So these screws look something like this, like the figure given here. Whereas thermometer is basically a device that you use to measure temperature. So like glucose meter and other pregnancy kit, etc. So these are not biomaterials. Please keep in mind these are external devices, okay? These cannot be classified as biomaterials. Now, this is another question that I have for you. Uh, what is a biomaterial? Okay, so take your time and maybe one of you can answer.
C.
C. C.
C, you're correct. So option C is the correct answer. Biomaterials can be either natural as well as it can be synthetic origin. It doesn't necessarily have to be a polymeric material. Also, it doesn't have to be a natural material or it doesn't have to be only synthetic material. Both can be, both types of materials can be classified under biomaterials. So if we just quickly look into what are natural materials and what are synthetic materials, so the natural materials we can again classify whether it is a protein or it is a polysaccharide. So under protein we have collagen, gelatin, fibrin, albumin, sulfibron, right? Whereas under polysaccharide biomaterials, we have materials like hyaluronic acid, alginate, chitosan. So these materials can either come from an animal or plant or bacteria. Okay, so since they are derived naturally from organisms, so that is why they are called as natural biomaterials. Whereas synthetic biomaterials, which are man-made, you are making it in a lab or an industry level. So you can classify it as again further classified as metal, okay, ceramic, then the polymers, there are synthetic polymers and semiconductor materials. So these are also biomaterials because these materials come in contact with the biological systems. So under metals, just like I showed you, the screws, those are of metal origin. Then the filling used for putting in your teeth, dental fillings, these are synthetic. Then the polymers that you use for making drug delivery systems, or if you want to make synthetic skin, okay, or cartilage, synthetic cartilage. So these are made up of various kind of polymers. Whereas for ceramics, people use ceramics to make again for dental application, then for bone orthopedic application, as well as heart valves have some ceramic component in it. So briefly, these are the natural biomaterials and these are the synthetic biomaterials. So this is the next question for you guys. You have to correctly match uh, column A to column B. Okay. So what is a viable biomaterial? Like, what do you think A matches correctly with which of these options in column B?
Tissue engineered muscle graft.
Okay, so one of you have answered that viable biomaterial is a tissue injured muscle graft. Then the option B, inert biomaterial. Again, what do you guys think will uh, closely match with all the four options? So if you think that more than one option is correct, so that is not the case. Only one will correctly match with another option in column B.
PLS suture.
PLS suture. Anyone else would like to try? Don't
Option
be
1.
option one. Okay. So you have answered correctly. Inert biomaterial correctly matches with option one. What about the C option, natural biomaterial, to which it
Fourth.
fibrin
Fibrin
glue.
glue.
Correct. So these leaves us to match option D with third option. Okay. So let's see if we are correct or not. So we are correct. So let me just briefly explain what are these four types. So natural biomaterial, you already know, I have already explained. But what are bioinert or inert biomaterials? So these are those materials which in principle will not interact with the host body. Okay. Um, that will also mean that it will not cause any um, positive or negative reaction to the host. Okay. Or it will not show any host response. So these are bioinert materials. But in practicality, any foreign material, so in all the biomaterials that you put inside the body, these are all foreign origin, right? So all of these materials will have some host response, means host will try to uh, elicit some kind of response. It can be very harsh one, it can be very mild one. So bioinert materials will then fall into those materials which gives a relatively milder host response. Host doesn't try to in the host you don't see very high inflammatory response, means all the inflammatory cells, the macrophages, neutrophils, etc., they like give a response, but the level does increase, but it gradually over the time it reduces. Okay. So these are bioinert materials, whereas viable materials are those materials which contain living cells. So tissue engineered scaffolds, since these scaffolds are made with cells or cell lines, stem cells, so that is why these are classified under viable materials. Whereas biodegradable materials are those materials which has the ability to break down into smaller, simpler compounds over a period of time. Now, these degradation, it can happen in response to various enzymes like proteases, or various kind of carbohydrate reactive hyaluronidase, etc. These kind of enzymes, they can act on it, or it can corrode out. Okay, so it can break down into smaller pieces on its own due to the surrounding environment, like maybe due to flow of blood or due to the activity of cells. So it can get degraded by that mechanism, and ultimately whatever byproducts that you get as a result of this degradation, these byproducts are usually or supposed to be non-toxic to the host. So if it is a toxic one, then we generally don't prefer its application for any particular biological application. So in the previous slide, where we were doing this question, the PLA sutures or polylactic acid sutures, so these are biodegradable or absorbable materials. So people have reported that they degrade degrade into simpler compounds like carboxyl, CO2, water molecules, etc. And these types of molecules can get easily expelled out from the body without any problem. And fibrin glue. So fibrin is another type of natural material which is naturally found in animal tissues. Okay. And in response, so it actually, this precursor from is fibrinogen, and in response to an enzyme called thrombin, it converts into fibrin and becomes a gel kind of a structure. Okay, so it's a natural material. Whereas PEG stands for polyethylene glycol. So these type of materials, these are polymer materials first of all, and they these to this material, the cells or the blood plasma, etc., they don't adhere well on the surface of PEG, which is why the cell and the material interaction is reduced when you coat any type of nanoparticles, say, with the PEG layer. Okay, so PEG will prevent the interaction between the cells in the host body and the material itself. So then the nanoparticles is then free to circulate inside the body and do its intended action, like maybe to deliver a drug or some DNA or RNA molecules or any other function that for which you have introduced their nanoparticle, which is why people usually quote a PEG or polyethylene glycol over these kind of materials because it makes the material, overall material, bio-inert, okay, non-responsive to host. Mm. So now let me ask you another question. So if you guys have watched the videos, then you will be able to answer. If not, don't worry, I'll try to explain this concept in as smooth language as possible. So what is biocompatibility? Okay, so I've given you
The ability
four.
of a material,
Yeah, can you repeat?
the, the ability of a material to not does not harm to the living being which is being implanted with it.
Okay, so you're kind of close to the actual answer, but let me emphasize on the fact that the biocompatibility concept is not very, yes or no kind of a thing, means black and white. It is not like that. It falls in a gray area. So, based on the results, we can say that it is biocompatible, but when you compare other results, then its biocompatibility can reduce. So that is why we say ki one thing is more biocompatible than the other. Okay, or this material is less biocompatible. Okay, so this kind of statements you can give when you clearly understand what biocompatibility is trying to tell you. So so you're kind of close, okay? You're kind of close. But maybe try to answer these four questions that I have for you. The first question I want to ask is there is a novel kind of urinary catheter that you have made, but this catheter has shown that its surface is very much susceptible to biofilm formation. So in that case, would you say that this catheter is biocompatible?
Not in that case. It
Yes,
won't be biocompatible.
yes, you're correct. So it is not a biocompatible because it increases the risk of bacterial infection, then ultimately will cause harm to the host.
Inflammation.
Yes, yes, you're correct. Then the next question that I have for you is you have made a drug carrier, okay, any carrier, so it can be a nanoparticle or a liposome, etc., and you have coated it with PEG, and you're using this carrier to deliver a drug. But unfortunately, this drug is getting delivered to a non-specific tissue. So you wanted to deliver your drug to heart, but it is going into liver. Okay. So in that case, would you say this particular drug carrier is biocompatible or not?
No, not again. Not again
No,
biocompatible.
not
Yes, you're correct. So, based on the results or result of this experiment, you will again say that the biocompatibility has reduced. So, or it is not at all biocompatible. But again, you have to check to how much whether it is causing adverse toxicity to liver or not. So if it is not causing effect on the liver function, then maybe you can say that it is less biocompatible. Okay, you cannot. So again, the gray zone. Okay, you have to say how much damage is there. The damage is less, then maybe we can stay. Still say it is a good material or a good drug carrier because it doesn't cause too much harm. Okay, but if it is stopping the liver function and ultimately causing a very bad systemic response, then in that case we cannot at all say that the material is biocompatible. And the third question I have for you is so you have made a artificial skin graft, okay, with cells and all, and you're observing from your experiments that it is able to accelerate the wound healing process. Means it is healing your wound faster. But after the healing has taken place, like maybe a month or two, scar formation has taken place. So in that case, would you say that this particular skin graft that you have made, is it um, biocompatible or not? What would be your answer?
No, maybe
less
partially
biocompatible.
biocompatible.
So again, the answer can be either yes or no. So if your objective of the study or is to just accelerate wound healing, then in that for that application, this skin graft works very well. Okay, it has very high biocompatibility. But you, if you also wanted to give some aesthetics, cosmetic aesthetics means good looking. So if the scarring takes place, then that particular area doesn't look good, right? So if your objective, main objective was to make it a very suitable graft so that the damaged area after it heals becomes very natural looking, very nice looking, no scarring at all. So if that was your application in the beginning, then for that case, your this skin graft that you have made is not biocompatible. So it's a yes or no, depending on what was your initial objective or what was your initial application for which you have made this biomaterial. Then the fourth question, you have made a knee joint prosthesis, okay, and you have tested an animal model for years, and you saw that after 2 years the effectiveness of the knee joint prosthesis is not anymore, basically it has failed. Okay, so in that case, would you say that the material is biocompatible?
No.
Okay,
No.
okay, so um, in this case, I think it would be a clear no, because these kind of processes are meant to survive throughout the lifetime of the patient. Okay, it cannot just fail in 2 years, um, because then the person, whoever has this particular prosthesis, has to undergo the um, what do you mean, the surgical thing, and again the pain will be there, the quality of the life of the patient will get compromised. So for this particular application where the prosthesis is meant to last for the lifetime of the patient and it has failed within 2 years, then the biocompatibility is not there. We have to look for any other type of prosthesis for this application. So given that you have understood from this example the general idea behind biocompatibility, maybe this definition of biocompatibility will make sense to you. So biocompatibility is the ability of the material to perform within a appropriate host response in a specific application. So in all these examples that I gave you, there was some intended application, right? And for that application, we had in mind some appropriate host response. If that host response was not completely fulfilled, then the material is not at all biocompatible. If some partial degree of fulfillment is there, means some level of appropriate host response was seen, then we can say it has low degree of biocompatibility. But if it is like not at all good, okay, not at all showing our desired response, our desired host response, then we should discard it and try to engineer our biomaterial again, so that we can again try to think that our material shows the appropriate host response. Now again, I have a question for you. Can a biomaterial exhibit biocompatibility across all the host into which it is implanted? Simple yes or no. Would suffice.
No. It
You
will
are
No.
depend
correct.
No.
upon the hosts into which
Mm,
it is implanted.
yes, you are correct. So base we have to then if say for the example number 4, where the knee joint prosthesis failed after 2 years, we cannot again say it is not at all biocompatible by looking at only one subject. We will have to look over a population of say 100 or 1,000 of people and then draw a conclusion that overall in a population of 1,000 people, how uh, frequent, like within how many years the prosthesis failed. If it failed within 2 years in all the 1,000 people, then it is not at all biocompatible. But it is only the case in 1 or 2 people, then we can say ki yes, it is biocompatible, but again the but will come because it has like didn't function well in 2 patients. Then we have to go back and do some research why it failed, what happened to the knee prosthesis. Okay, we have to do some additional investigation so that we can make sure that in more and more people our material, our biomaterial gets accepted very nicely and gives the appropriate host response for a specific application. Now, this is a very simple question if you have understood the concept of biocompatibility. Select the statement which correctly relates to biocompatibility. First,
Option
yeah,
number C.
option number C. What about others? Does anyone agree with option number C, or is it just one person?
Uh, all the
Option
options.
C.
Yes, you were saying something?
Yes, all options are like more or less appropriate, but the C one is the most appropriate one.
Yes. So those who have answered, you have answered correctly. The correct option is C, because here it is much more general. Okay. So the option C should be sufficient. Like it should, the biomaterial should stick to option number C, or it should show the property of option number C. After that, we can say that it should have the exact dimension, or it should have therapeutic characteristics or healing characteristics. But option number C, it should be like the primary thing when you are trying to say whether your biomaterial is biocompatible or not. Okay. Now, this is another question for you. Do you know which regulatory body is responsible for approving whether this biomaterial should come into market or not?
FDA.
FDA. You're correct.
FDA.
Yes. So it is the United States Food and Drug Administration that is given this task, and all over the world, okay, we follow the FDA approval. Even in India as well, there is no other body in India that does this individually. Everyone blindly follows that FDA is approved. So yes, we can use that in clinical setting. What are the what were the other options? Like what were the other regulatory bodies? So FASI, FSSAI. So it is the Indian Food Safety and Standard Authority. It makes sure that whatever food that comes to our plate is safe to eat. Okay, so it makes sure of that. Then BIS is the Bureau of Indian Standards. So this particular body makes sure that each and every product that comes into the market meets some certain standards. Like even if so let's say the sutures, okay, which the surgeons used for stitching. So those sutures for using in the hospital setting, it has to fulfill from some BIS given standards before it can be used in the surgery. If that particular product didn't fulfill, then the Indian government won't allow its application in the hospitals. Then ICMR is basically is a body that makes sure that medical research goes on over the country. So there are various ICMR approved institutes, okay, where research on various kind of diseases and as well as cancer and biomedical research goes on. Okay, so they don't necessarily give some regulatory decisions. They give research to help the regulatory authority to make laws and rules. Now, which one is not a dental biomaterial? Cement, amalgam, polycarbonate, and TI stands for titanium.
A.
Option A. Okay. Anyone else would like to try?
C.
Can you repeat?
Option C.
Option C. Okay.
Polycarbonate.
Polycarbonate. Okay. So those who have said the option C is correct, you guys are correct. The correct answer is polycarbonate. So the cement, amalgam, these are used as dental filling materials. Okay. Whereas titanium, they are used to make dental implants, and they're used to replace some missing teeth or damaged teeth. Whereas polycarbonate is a material which are mainly used to make goggles, like lab safety goggles, sunglasses, etc. So for the eye protection externally, um, whichever sunglasses you wear daily in your life, they are usually made up of polycarbonate, because they have the property to protect our eye very nicely from UV, etc. Then another question I have for you. Orthopedic implants are not made of titanium, SS316, that is stainless steel 316. HA stands for hydroxyapatite, and amalgam.
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 Amalgam. So you guys are correct. Uh, Amalgam cannot be used to make orthopedic implants. Why? Because it is much softer, means is overall Young's modulus value is much lesser than the other metals that are used for orthopedics. So uh, these types of here in these are the types of implants used for orthopedic applications. Like you can, so here is the hip total hip joint replacement. This is how it looks like. Then other plates, bones, screws. So these can be made up of titanium, stainless steel. Whereas hydroxyapatite, which is a type of a ceramic material, it is used as a coating, okay, on the surface of these metal implants. So hybrid type of prosthetics are being now made because hydroxyapatite has its own advantage over metals. So if you know the structure of our bone, hydroxyapatite or the calcium phosphates, they are one of the major constituent of the bone. And in order to make the biomaterial much more acceptable to the host, people have started coating it. People have observed that the cells like the osteoblasts, etc., they respond better to the prosthetics because they can easily adhere on the substrates and start mineralization process, and start making bone, like bone regeneration. It helps in bone regeneration when you use this kind of hybrid prosthetics. So if you guys have any queries, okay, you can freely unmute yourself in between, interrupt me in between and ask, because you guys don't have the chat option visible in your portal, like in your window. So that is why you can easily mute and speak. Then in 2015, which is the largest biomaterial, sorry, second largest biomaterial market? Orthopedic.
Orthopedic.
Okay, orthopedic. Other options were cardiovascular, dental, and ophthalmology. So in 2015, orthopedic was the largest biomaterial market. You are correct. But let me just show you this data, which I've obtained from this website, Grandview Research. And in this data, this is the current data, 2022 data. It is very current data. And here, what I've read is uh, orthopedic, although in 2015 it was second largest, but currently orthopedic is the largest biomaterial market. Means in the market, more and more orthopedic implants are available. Whereas in 2015, it was the cardiovascular implants which captured the largest market size. And now it is kind of reversed. And also, if you look at the figure, how big the global market size has become, earlier people projected that it should be around 130 billion US dollars by 2020, but this field is expanding so much. Okay, in every country there are hundreds of labs who are doing research in biomaterials. More and more companies are coming into this area and willing to work with the research labs and help various kind of products come into market, which is why the market size has grown a lot. It is now around 155 billion US dollars. Okay, so if any one of you has interest in this field and want to work, you can easily find opportunities is either in research labs or in industry. Okay, maybe start your own startup maybe. Now, another question, the nanocomposites. Okay, what generation of implants are these? First, second, third, or fourth generation?
Fourth generation.
Fourth generation. Okay. Anyone else would like to try?
Third generation.
Third generation. Okay. So, how are you able to answer this question? Just have you memorized it, or it's just a guess?
In the beginning, you said about the fourth generation implants while starting the lecture, so that's why I recalled it.
Okay. So the correct answer in this particular question is third generation. Why? Okay. So let me just briefly show you this particular slide. So the if you look over the time span, how the biomaterials have evolved from 1950s to now, beginning in the beginning, people were more interested in those materials which showed the property of bioinert. They didn't want those materials to elicit host response. Okay, that is why materials like the metals, alloys, so these were considered as bioinert and had people used more in, and they were easily FDA approved for clinical uses. Later on, when people started to realize that some degree of host response is necessary, but we can tune that host response, how the host is reacting. So that is why then the second generation material came, which were bioactive, means they were able to elicit a specific kind of host response, or these materials also had the property to degrade inside the body, because people thought patient won't have to undergo second surgery to remove that material. Rather, it should degrade inside the body, which is why various ceramics and polymers came into picture, and their use increased and gained FDA recognition. Then materials like nanocomposites. So these composites materials are basically metal, polymer, polymer ceramic, okay, or ceramic metal. So these are hybrid kind of composites. Those kind of materials they came into market because individually when you look at ceramics and polymers, they may not have the appropriate amount of strength, or they may not be able to fulfill or mimic that particular tissue area. Okay, or if you see at metals or alloys, they have a very high strength. Okay, and the response that people have obtained inside the body is not very desirable. Like either people are seeing some high level of fibrosis, like more and more fibrous tissue is getting accumulated, which is why the activity of the material is dishampered. So in order to combat this kind of limitations of either of these, people have started making hybrids, okay, like polymer metal, metal ceramic. And when they observed how they were working, they saw that it is doing very good. So I just showed you one example where hydroxyapatite was coated on the surface of the metal so that it gets better accepted inside the body. Ultimately, the idea is the aim of a biomaterial scientist is to make such a material that gives the most appropriate host response. If the host is showing some adverse effect, then the material biocompatibility reduces. So because of that objective, we are changing the design of our material. Okay. And the last type of materials, like the current type of materials that we are working, like I'm working in the field of tissue engineering. So the biocompatibility of these kind of materials further are increased because people are using cells directly from the host, or they are using various kind of materials which are originally found in the body, like collagen, fibrinogen, etc., or they're using some growth factors. They are again present in the body, so that the acceptance of that particular biomaterial and ultimately the degree of host response that has reduced. So that is the uh, fourth generation biomaterial, where people are trying to mimic the native tissue and ultimately increase its biocompatibility and increase the desired level of host response. Okay, so in the previous question we were discussing what are nanocomposites, or you can say the composites. So these composites, they come under third generation of material, but the tissue engineered scaffolds, they are the fourth generation material, and these are the very recent materials where which is we are currently looking at. Okay, so this is another question. Which which among these four polymer has a chlorine group present in it?
Option number D, PVC.
PVC. Okay. So that is relatively simple if you already know the structure. So let me just show you how these four type of polymers look like. So this is the PVC structure, where the chlorine atom is there. So, and these are since polymers. So in this figure, I'm showing the structure of one monomer, and the N represents how many of these monomers will be like forming a particular polymer molecule. So PVC, it has a chlorine group, whereas PTFE, also called the polytetrafluoroethylene, or its commercial name is Teflon, it has all fluorine groups. Okay, so here two carbons are there, and three of them are hydrogen attached to it, and one is chloride. But in PTFE, all of them are fluorine. Then in PVA, if you see again, the similarity in structure is there. Only in place of chlorine, there is a OH group, alcohol group is there. So this is how the monomer of PVA looks like. And if there are like N say 120, so this will be a very long chain. Then whereas in case of polypropylene, which is basically a type of a plastic, here the OH group, again the structure is very similar. See, two carbon is there, hydrogen. Okay, so three hydrogen groups, but in place of one there is one methyl group. Okay, so that is why so this is now become the polypropylene. So this OH group is here, this is methyl group is here. Here all four groups are fluorine, and here only one group is chlorine. So depending on how the monomer looks like, people have given the name accordingly. And the poly stands for the polymer, okay? And N stands for how many monomer groups will be there in a particular polymer. And if we change the number of N, I forgot to mention, if we change the number of N, we can tailor the molecular weight of the polymer and ultimately its physical and chemical properties, okay? Mechanical properties, etc. We can tailor it accordingly. Now, this is again a match the following type of question. Here you have to correctly match one option. There are three they are there in two columns, distributed in two columns. Just have to simply match it. So let me ask you, which of these ABC correctly matches with metals?
The first one, highly tailorable physical and mechanical properties.
Okay. Um, Anyone else would like to give it a try? Okay, so metal, the one unique property of the metal is it has a very high Young's modulus. Okay, and it has a elastic property. Because of this elasticity, it gains a property called as shape memory. Means what is shape memory? So if the metal is under some load, okay, and it because of which its length has increased. Now if you release that load, it can again go back to its original shape because of its elasticity, and thus it has a very high shape memory property. Okay, now now we are left with two more options, A and B. Which of these correctly matches with polymers?
A.
Yes, you're correct. So I've just mentioned. So if you are making synthetic polymers, like in your lab, you can basically change the number of monomers and you can get a different mechanical property or different physical or chemical property. Similarly, if you are using, say, natural polymers, means the polymers derived from natural origin, depending on what species, from which kind of species you have obtained, its property again changes, but the tolerability in case of natural polymers it slightly reduces because you have that it depends on the species. You cannot change the species, right? But you can still fine tune its property if you add some chemical groups to it. So those are called semi-synthetic polymers, like in between. And these are again a new class of polymers coming into literature, like in very recent years. Okay. And the last option that we're left with, ceramics, these have a low tensile strength. So let's see. Yes, we are correct. So briefly, let me uh, like compare all the three synthetic biomaterials classes: polymers, ceramics, metals, and what are its advantages and disadvantages. So the polymers, it has various good advantages that is good for a particular application. So you can easily fabricate using polymers. So you can use 3D printing, you can use electrospinning, you can use freeze drying or solvent casting, porogen leaching. These are all type of techniques, and this technique specifically uses polymers to create various kind of shapes and geometries, and that you can use for a particular application. So the fabrication step becomes very easy if we are using a polymer. Then yes, physical, mechanical, chemical properties, these all can be fine-tuned. But exception is if you are using a natural polymer, then the degree of tunability reduces. Then surface modification is possible with polymers. You can make the surface very hydrophobic or hydrophilic by using different kind of treatments, of chemical treatment, like you can covalently bond some functional groups, or you can do some physical treatment like uh, do oxygen plasma treatment, or you can either also use some biological agents. Okay, you can allow cells to adhere, because of which the surface property of the biomaterial has completely changed. So the surface modification, also why surface modification is important, it is because it is the surface of the biomaterial which the host cell sees first, and accordingly it will decide whether what kind of response to give. So if the surface is modified in such a way that the host cell recognizes as a foreign, but let's see, the degree of inflammation that it gives in response to a foreign object is not that high, the biomaterial property is able to reduce that inflammation degree, then its biocompatibility increases. But if it is other way around, inflammation high happens to a very high degree and it persists for a very long time, because of which other effects like bacterial infection, etc., they starts to happen at the biomaterial site, then the surface modification that you have made is pointless. Okay, its biocompatibility decreases. So we have to learn how to do surface modification and ultimately do experiments to see its effect. Then there are those kind of polymers which are biodegradable in nature, and biodegradable polymers are good because surgeons don't have to do a second surgery to remove it from our body. Then again, we have to study what is the toxicity effect, if there is a toxicity effect of the biodegradable byproducts or not. If these are toxic, if these biodegradable products are toxic, like uh, case of magnesium alloys, people are studying this kind of biodegradability studies, and they're showing that some ions, some metal ions that get released overall, at the local site as well as systemic site, they are studying how toxic it is, and if the toxicity level is low or high. Okay, so that will again decide the biocompatibility. Then polymers can be used to immobilize cells. So under polymer, there is a subclass of material called hydrogels, okay, which we will discuss maybe in subsequent weeks. These hydrogels, they are a type of polymers which have the property to retain a large amount of water in it and create a aqueous environment. So as you know, the cells that are present in our body, they are always present in aqueous environment. So these hydrogels can be used to mimic a aqueous environment that is present inside our body, and because of which cells in within the hydrogel, they thrive well, they grow well and show various kind of biological properties. Whereas there are various disadvantages to polymers. There are some polymers who leach out compounds. So leaching is a mechanism through which the uh, polymer biodegrades. So if that leached out compound or leached out molecules, if those are toxic, then the polymer is not at all good. Okay, then surface contamination is possible. So if the surface during the surface modification, and if it has happened uh, in a non-sterile environment, okay, even one bacterial cell, if it has adhered on the polymer surface, then it will then go inside the body and ultimately can result in biofilm formation and bacterial infection. So it is very, very crucial um, to sterilize to optimize the sterilization process, as well as to make sure that the surface modification doesn't allow this kind of phenomena to happen. Then wearing and breakdown is has been seen with polymers, especially in those polymers which are intended to stay in body for a very long period of time, and at locations where the bones where location where constant flow of fluid is there or movement is there. Okay, so in those type of locations, wear and tear has been observed. Then for some application, biodegradation is not necessary or is not desired. Say for tissue engineering application where you are making a scaffold out of polymers, and for an application, say wound healing. So we have to fine tune this biodegradation rate with the normal healing rate of our body. If the polymer has degraded before the wound has healed itself, then in that case, the use of this polymer is disadvantageous to us. We have to make sure that the degradation rate of our polymer is equivalent to the healing rate of the tissue, so that over a period of time we will see more and more host cells, they have uh, reformed that injured site, and the polymer that you have used is no more present at that site. So you have to like basically a trend kind of a thing. In time zero, the polymer was high, whereas in time zero, the host cells at the site is the lowest. But when say you are doing the study for 30 days or 2 months, okay, so in that case we should observe if the equal rate of degradation and the rate of healing is equal, then we should expect that the host cell at the site is the highest, whereas the polymer content is negligible. So if that is possible, then only it is advantageous. If that is not happening, then the biodegradation property is disadvantageous to us. Then there are often various kind of polymers that doesn't respond well to sterilization mechanisms. So we usually use methods like germicidal UV, that is 295 nanometers of UV. So that can kill the bacterial cells. Then there are other methods as well, like gamma ray radiation. Gamma ray is used, then ethylene oxide gas is used to sterilize. So these three are the most commonly used sterilization process. And another is autoclaving. So you use very high pressure steam to kill all the bacterial spores, etc. So if your material, if your polymer doesn't or it degrades in presence of these kind of harsh treatments, or its property, say mechanical strength reduces, or if some chemical reaction has happened to it, or some surface contamination, as I told, some has happened because of sterilization, then we won't be able to sterilize it well. And if we cannot sterilize it well, it is not at all safe to introduce inside a human body. If, now, what are the advantages and disadvantages of ceramics? So ceramics, like hydroxyapatite, aluminum oxide, etc. So these materials, they have very high compressive strength. Compressive strength means you are able to compress it very like they don't break. They don't break that easily when they are under a compressive load. Then they show a very high corrosion resistance and wear resistance. So when some movement of fluid or bone, etc., is causing corrosion on the ceramic material, then uh, layer by layer, if the molecules and the atoms, they are getting eroded, the below layer getting exposed, right? The bottom layer is gets exposed, and and over the period of time, if that is happening, people observe what is the final property of that particular biomaterial. Is it good? If it is almost same as the original one, then in that case, you will say that the material is showing a very good resistance. Okay, but corrosion, wear and tear, these kind of effect will always happen. Okay, and it also depends on what location your biomaterial is inside the body. So you cannot like minus that particular factor, but we have to see the effect of that factor on the material. Then the ceramics, they can be highly polished, they can be made very polished. So various kind of cells inside our body, they require a very smooth surface for adhesion. Okay, otherwise they will not show any surface adhesion on the cells, and ultimately whatever response you want to observe, that won't happen. Then ceramics can be made in such a way that it is bioactive, like you can maybe load some drug or allow cell adhesion on it. Okay, that is, you can um, basically direct the host response, or there are various kind of ceramics which are bioinert, that doesn't get affected by adhesion. So orthopedic and dental implants. So the strength, it should like the strength of the ceramic, it should match with the bone or the teeth. Then the effect people have seen is better. If it is either way, then the effect is not good. Means healing or whatever application that you are using this material, you will won't get very good results. Tensile strength is very less. Even though it compresses very well, it doesn't respond well when you are trying to pull it from both the sides. Means you are giving some tension. It breaks very easily, which is why the next point is valid, that it has low Hello, am I audible?
Yes, now you are audible.
Yes. Yes. Actually, in between, my internet connection went off because of which I yeah. Mm. So is my screen visible?
Yes.
Yes. Thank you. So where was I? I was trying to explain that ceramics, the ceramics material, they don't have a very good tensile strength. Means if when we pull ceramic material, it fractures very easily. That is why ceramics are very brittle. Okay, and using ceramics, can you please unmute yourself, Nitin? Can you please? Yeah, thank you. So, and also using ceramic, it is very difficult to create complicated type of structures. Okay, whereas for using polymers, various kind of easy fabrication techniques are there, and you can make various kind of nice complex architecture. Whereas for ceramics, it is difficult. Now coming to metals, metals like ceramics, they have a very high strength, they have very high Young's modulus, but again, uh, sometimes metals are having a Young's modulus value much higher than that of the native tissue like bone cartilage. So because of which the biocompatibility of in metal individually reduces. People are trying to make hybrid materials like some metal with polymers, etc., so that you can match the strength of the biomaterial implant more closely with the target tissue. Metals are fatigue resistance. Okay, so when we'll talk about mechanical properties, we will maybe discuss more in detail what is fatigue. But for now, in brief, fatigue is basically um, a property of a material, okay, where in presence of cyclic load means you have a metal bar and you are continuously stretching it and then releasing it, stretching and releasing it. So there is a cycle going on. And in presence of this cycle, inside the bulk of a metal bar, some cracks starts forming, minute micron size cracks. Okay, and people study how many such cyclic loads are needed for the material to break. So for metals, people have observed that uh, it will not at all break even if you give a very high number of cyclic load. Okay, that is why this in that case the statement like it has a very high fatigue resistance is appropriate. But if it breaks quickly like in 5 or 10 cyclic loads, then the fatigue resistance is very less. And fatigue resistance, why it is important to measure? It is important when you are using this type of metals in sites like a pacemaker or valve, okay, heart valve, where continuously the movement is there, like opening and closing, okay, or any other body part where this kind of cyclic load is experienced or will be experienced by the material. Then the wear resistance is there. Okay, then again metals, given that it has a very high elastic property, you can use various kind of techniques like 3D printing. By the way, you can 3D print metals because again you can melt it, bring it to liquid state, or you can stretch it. Okay, has you can make it into wires. The fabrication process in for metals is very easy. For hundreds of years, people have perfected the fabrication techniques for metals, and that makes our job easy, or to make various kind of structures of small to very large sizes of complicated shapes using metals. Sterilization process is also easy. Uh, Usually, the surface of the metal is inert to the sterilization agents that we generally use, whereas for polymers some kind of modification happens. Okay, and because of high elasticity it has very good shape memory. But the one major disadvantage is this metal ion sensitivity or toxicity. So what is metal ion sensitivity? If you just simply search the internet metal hypersensitivity, you will come to know that it is a kind of an immune response, a very common immune response that people have observed in the patients having some metal prosthesis. Here what happens is continuously um, the macrophages or the neutrophils, they are activated. They're activated, and they start to release various kind of cytokines at the site of the implant. Okay, because of which the inflammation process basically elongates. And the effect, the local, it is the local effect which I talked about, but there are some systemic effects that also comes into picture in the human. So that person develops some allergic reaction, like unwanted antibody starts getting created. So in some cases it can be simple, like you can treat it with some medication, okay, and you can reduce that. But in some cases it can becomes a life threatening thing as well. Okay, so people have to study in that cases that the ions, predominant metal ions that are there in the prosthetics, what effect does it have on the immune response. Okay, so these kind of studies have to be done before metal based implants get approved. Then toxicity is also there. So some kind of ions can get into the cell, hamper the mechanism of the cell, and again hamper overall function of the tissue. So these have to be studied. Then only some biocompatibility can be said, whether it is biocompatible or not. Now, this is a question based on the concept of uh, stress and strain, then Hooke's law. So this is a question on strain. Okay. So you are given a piece of polymer, and this polymer is extended by a force that changed its final length to 5.5 centimeter. Means it basically uh, increased. Okay. It's increased its length because its original length was 5 centimeter. You have to calculate what will be the, or what is the amount of strain experienced by this piece of polymer. So if you, sorry, I was not going to show you the answer, but no worries. I have to show you how did we come to that particular answer. So if you have like gone through the lecture, you know that strain is basically a relationship which is the change in length by the original length. So stress is a load or the external force that you have given divided by the cross-sectional area on which the force is applied. So now note this strain and stress, these two quantities are normalized quantities. Okay, so if you take a circular kind of a metal implant, and if you take a cylindrical shape, and you want to compare the effect of force, the change in length in both of these geometries, you won't be able to do until you use these normalized relations. So the amount of force here, and if you compare the amount of force experienced by the circular geometry, these two will be completely unrelated to each other. Until and unless you calculate the stress experienced by this geometry and the stress is experienced by this, then only you can compare. So these are normalized quantities irrespective of how the final geometry of the material looks like. Now, in our previous question, we were asked to calculate strain. Okay. Now, we were told the final change in length, which was 5.5 centimeter. Okay. And the original length of that polymer piece was 5 centimeter. So from this, we can calculate what is the change in length. So it will be 0.5 centimeter. Then simple strain we can calculate, and it is represented by the Greek letter epsilon, and you can simply take a ratio of 0.5 divided by original length, that is 5, and then the answer will come out to be 0.02. As you see, the numerator and denominator both have the same units, so it is a unitless dimension. Okay, it is unitless. So were you able to do it yourself, or did you have any query while I was doing this problem in front of you? Anyone, any query? So in the previous, yeah, any query?
No queries.
No queries. So if suppose you guys were given the original length and simply given delta L, so you could have easily calculate the strain. Or the other way around, if the strain is given to you, value of strain, and you were given the, say, the change in the length, then you can get the original length. So if two things are given, you can find out the this third quantity. Now you also have to like understand from the question what kind of strain it is. So if say this is the object and the force is applied outward, so the change in the length is in this direction, correct? So this is one strain. But if you but the effect of this longitudinal or this outward force can be seen in this direction, opposite direction as well, like perpendicular to it, which can reduce the diameter of the cylinder. So then the value of strain will become something else. So this is strain 1, this is strain 2. So these kind of thing you have to understand as well from the question, what kind of strain it is asking, and accordingly do the problem. So I think this is the correct answer, 0.02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. 02. And
Ma'am,
this is another, yeah,
ma'am, why it is 0.02? It should be, I think, 0.1.
should be 1. Okay, you're correct. You're correct. Should be 1. Yeah, point 1. Yes, you're correct. Thank you for noting it down. Okay. So this small length, the denominator should be the original length, not the final length. In that case, you are correct. Yeah. Anyone else have a similar doubt? Is it clear?
Yes, ma'am.
Yeah. Thank you. So someone was listening. That means listening actively. Very good. So I really appreciate it. If in future, if I make any kind of mistakes like this, please feel free to like stop me in between and correct me. Okay. So the correct
Okay,
answer
ma'am.
is not this. This is point. Okay. Okay. This is another problem based on stress strain and then elastic modulus. So in the previous slide, I talked about stress and strain. And when we take the ratio of stress and strain, this is the stress. Small sigma is used to denote stress, and this is epsilon. This is strain. That comes out to be a modulus called Young's modulus. So I told you that stress and strain, these are normalized quantities. Okay, but in when we try to compare in between different type of materials, okay, then the inherent property is the Young's modulus. So every kind of material, whether it is a polymer or a metal or a ceramic, has a inherent Young's modulus value. And and in that case, we cannot compare stress or strain values. Stress and strain values are comparable when you are talking about different geometries, but the both two geometries should be made up of the same material. Then only you can say some arguments like yes, stress, this material has experienced more stress. Okay, but Young's modulus is an inherent property of a particular type of material. Now let's just briefly look at the question. This question is talking about a guide wire. So this is having a cylindrical geometry. The length of this wire is 4 meter. Okay, and the diameter is 5 mm. Okay, or maybe I will do it here. Okay, I'll do it on this slide better, really better. Elastic modulus is this value, and this is just writing down the formulas for you and for me as well, so it becomes easier. So if we briefly look at the question, uh, from the question, so can you unmute, sorry, mute yourself? Uh, okay, fine. So I've muted the participant. So from the question, we know length of this wire, which is having a cylindrical geometry, is 4 meter, and the diameter of this wire is 5 mm. Now it is better to convert your problem into same unit. Okay, so if you look up, 1 meter is equals to 1000 mm. Okay, so we have the same unit of length with us. The force that this metal wire experienced was 20 Newton. And because of this force, there is some increase in length. And what is this increase? It is coming out to be 0.5 mm. All right. So we have to calculate the amount of stress, strain, elastic modulus experienced by this particular guide wire, and we have to assume that it is an elastic material, means it doesn't undergo any plastic deformation. Okay, plastic deformation means the change in length which is irreversible. The material cannot go back to its original length. That is called plastic deformation. Elastic means how much is the change in length, it will come back to its original length. Hooke's law, stress is directly proportional to strain. So from that, we know the material is purely elastic. So let me just calculate stress. What will be stress? Stress will be the force 20 Newton by area. Now this is the, so if this is the wire, okay, and the force is say towards only one direction, and this force is always perpendicular to area. So this is the area. Then the area will be simply pi r square, correct? And here is the force. So the area we have to then calculate, right? Pi, here diameter is given, so d by 2 is r square. So if you have your calculators with you, you can also do it along with me. So first, we can get radius 2.5 millimeters. Correct. Please do along with me if you have your calculators and pen and paper out. So the area, the cross-sectional area of this wire is coming out to be 19.63. Okay. So for getting the stress value, we just have to divide the force 20 with the area. And what we got is this 1.0188. So this is in Newton per mm. Okay, Newton per mm. Please mind it. So we can approximately write it as 1.02 Newton per millimeter square. This is the area unit, right? So we can convert it into the SI unit of stress. So what is SI unit of stress? It is basically Pascal, okay? And Pascal is Newton per meter square. So we have to basically then convert this mm to meter square. So how will you do it? You can do it like this, Newton. Now, this will, as I told you, 1 meter is 1000 mm. So 10 to the power minus 6 to this, okay, meter square. Let's just look, okay, if I'm doing correct or not. If you're also doing, you'll able to understand. If you're just watching, it may seem like I'm doing everything correct. It is Newton per meter square, or you can write it as, this is what? This is mega. 10 to the power 6 comes out to mega, and this Newton per meter square is our SI unit, Pascal. So the stress value is this. Okay. So we have solved one part of our problem. Let me just do the another part. So we have to also determine the strain value. Again, just for reference, I'm writing the strain formula from the from this particular question. We understand that this question is talking about only strain in one direction, okay, in only one direction, in the longitudinal direction. So we'll focus only on that. So the original length of the wire was how much? 4 meter, correct? And the change in length is given here. It is not given the original length, rather it is given how much by how much, what is the change it is given. So it is 0.5 millimeter. So better to write it in millimeter so that becomes easy for us to calculate the strain value. Here epsilon is strain. What it will become? 0.5 divided by 4000. So it is okay. So it is coming out to be 0.000125. If we simplify it, will come out to be 10 to the power minus 4. This is the strain value. Then strain, as I told you, it is unitless, so it will not have any unit. Now, last quantity that we have to calculate from this question is the Young's modulus, E. And it is basically the ratio of stress that we have calculated already and strain. Just what is the value of stress we have got? We have got 1.02 megapascal, correct? And the strain value is 1.25 10 to the power minus 4. Simply do the division and you will get the answer. 2 divided by this point, 1.25. Okay, so what you have obtained? You have obtained 8160 megapascal. Now, if you want to write it, you can also write it as 8.16 gigapascal. So now giga stands for 10 to the power 9, 10 to the power 9. So you can write elastic modulus to be this. So you see, for a metal, that whichever metal it is not mentioned in the question, the elastic modulus is quite high. It is in the order of 10 to the power 9. It is in the order of gigapascal. So this is how you would simply solve a question based on Hooke's law. Okay, where stress, strain, and elastic modulus you have to calculate based on the information that is given to you. Okay, so now in the next class, for the next week, basically, I plan to discuss more in detail the mechanical properties. Okay, there I'll go in detail the theory part. But for this class, I'll stop here only. And if you have any queries, feel free to share it. Okay, I can maybe go back to few more slides in the beginning for those who have joined late. Okay, and yeah, now the floor is open for questions.
Can you please show the previous slide?
Okay.
Stress calculation.
Yes.
Okay, ma'am.
Right. I hope you guys have got the same answer that I have. Or anyone is getting any other answer. See, the objective is not to show you what is the correct answer, but rather to show you how to think in order to get the correct answer. That thinking process you need to learn. Then whatever question is given to you, you will be able to solve. Doesn't matter which language it is in or how it is stated, you will be able to get it. Just you have to understand how to think, how to approach. Okay, then only you will be able to get the correct answer. Don't think कि ये just formula दिया हुआ है and we have to just put the values. It's not like that. Maybe the question is framed in such a way that you won't get the answer using a single formula. You have to look back and use multiple formulas in order to get one answer. Okay, so application of the formula is also um, like smart thing that you have to learn. So actually, I will discuss all the mechanical property concepts, okay, the Poisson's ratio, etc., whatever is there. And Professor Doble has discussed various kind of problems as well. So I'll also discuss similar problems. It will definitely help you in week 2 assignment, okay? But whatever content that I've discussed today is sufficient for week 1 assignment, okay? So I hope after today's class, you guys will be able to sit and solve the week 1 assignment within 10 minutes. Like it shouldn't be that difficult at all. For week 2, come prepared with the like view the lectures first. Okay, and if you have any queries, come prepared with them on the next Saturday, and we will discuss your queries, as well as we will briefly discuss the concepts as well. And that will definitely help you to solve the mathematical problems for week 2. Any queries from anyone? It's totally fine. You don't have to be
Hello,
shy.
ma'am.
Yes, yeah.
Ma'am, this polymer has a advantage and a disadvantage, a common advantage and disadvantage, which is biodegradation. So does this biodegradation rate can be controlled with any surface modification of material?
So what you can do, you can engineer your material in such a way. Um, so you can make a hybrid material. Okay, if you want to control the biodegradation, you can make a hybrid material. You can coat your material which has a say. So if your objective is to reduce the biodegradation rate, means make the material degrade at a much slower rate, then you can coat it with a material which has a very low degradation rate. Some like and the inner core of the material would be your bioactive material, but unfortunately that bioactive material has a fast biodegradation rate. Means, let me just again explain what I'm trying to say. I'm saying is, if your objective is say to make sure that the material doesn't degrade very fast, means reduce the degradation rate, then you can engineer your biomaterial or your implant in such a way that the outer layer that the body first sees is that material that has a tendency to degrade at a slower rate. Okay, and slowly it exposes your bioactive material. And what this will help you, it will help you to elongate the action of your bioactive material inside the body, until, and if the thickness of your outer coating will also depend. So if you want for a very long period of time your biomaterial should stay inside without degrading, then you will make a thicker outer layer. Okay, but if you want to see its effect only for a few months, then accordingly you have to adjust your thickness. Then once your bioactive material is observed by the body cells or the enzymes or whatever plasma proteins etc, then you know that within a day or two your bioactive material will come out from the system, and till that period you should be sure that what for whatever application you have delivered that biomaterial, that application is fulfilled. Okay, so in this way you can engineer, or you can say control the degradation rate. What can be other ways? You can chemically modify the material. Means while synthesizing that material, if you're using suppose a synthetic biomaterial, you can add such biofunctional groups to this polymer chain, or if you are making an alloy, you can make a metal alloy, you can use a combination of those alloys, which, so we can use a combination of chromium, cobalt, titanium, right? You make a alloy of these three metals, then you will increase the percentage of those that metal which has a low degradation rate. You won't keep magnesium. So magnesium, it degrades very fast in the body. So in that way also you can play with the composition of your biomaterial, like the primary composition of biomaterial, and ultimately effect is can be seen on its degradation rate. There can be other ways as well. I just gave you two very simple ways that came into my mind. Did that clear your query?
Yes, ma'am. Yes, thank you.
Yeah. Any other query? Again, those who have joined late, and if you want that I should like show you some of the questions. Anyways, the slides and the recording, etc., will be made available to you. Okay, so you can watch it later. But still, if you want, because we have some time in our hand, so we can simply revise few questions earlier. Will become easier for you then.
So, excuse me, ma'am,
Yeah.
so this interactive section is recorded and it is available. It will be available in the website.
Yes, yes. So
Okay.
I will be uploading it. Uh, I will be sharing all the details to NPTEL people. Now NPTEL will create a problem solving session, live session section in the portal. So when you log in to NPTEL, you will be able to see that problem solving session. But it may take some time from their side to reflect. Okay, so if you want to say watch it tomorrow because today I'll upload it. Okay, then what you can do, you can just simply Google Mauchandra IIT PMRF, like in the Google, just search and you will find my YouTube channel. In my YouTube channel, you will find the recording by tonight only, okay? And you'll be able to see it immediately because I'll be doing it instantly. And after this recording is done, I'll be uploading it. So by tonight, it will be available to you. Unfortunately, I'm not able to type it in the chat box because the chat box is not visible to you guys. So I can just show you the link. Save it. So this is the YouTube channel. Okay. Mauchandra PMRF IIT Kanpur. Just search in your mobile, in your Google, okay, and you will find this YouTube channel. So I've actually been a TA for another NPTEL course previously. So you will find a playlist of that recording as well. So this is the first class. So I'll create another playlist for all the lectures, all the sessions that we are doing for this particular course, and you will find the recordings under that playlist. Okay, and the notes and all will be made available to you as well. So since I'm not allowed to directly share it, then from the notes that you will get directly from NPTEL, okay, in the portal. But recordings you can find tonight if you just go to my YouTube channel. Okay.
Ma'am,
Yeah.
could you please clarify the format of final exam which will be on 24th March? Like, will it consist of subjective questions or only MCQ?
See, I actually don't know. So that is why I don't have a clear answer to this question. It totally depends on the professor, okay? As well as, see, I am a PMRA student. I'm studying at IIT Kanpur. So there is another TA for this course, and he is working with Professor Doble. So he and Professor Doble, they will decide what kind of pattern it will be. But based on my experience on giving, I have given NPTEL exams myself. So based on that experience, I can tell you that most of the questions will be MCQ type. Okay, but don't take it like very true. It is just based on my exam experience, which I've obtained by giving exams of other NPTEL course. Get it? So
Yes.
so usually what they do is they give you a center, okay, exam center. You have to go there and you have to give the exam in online. So in that case, it might be in the JE or format where fixed time will be there and you have to like correctly answer few MCQs. Maybe it will be like that. I don't know whether they'll be subjective or not. Or if it is subjective, hardly one line answer should be there, not very big answers, but it will be online form, that I can assure you. It will be an online exam.
Okay, ma'am. Thank you.
Yeah. So all of you have registered for exam, or any one of you is still thinking?
I haven't registered yet, but will register it before the deadline, before 7th of Feb.
Okay, great. So any other queries related to the week 1 topic or anything that I've discussed today? Okay, if not, then maybe we can say goodbye, and I hope to see you all I think today 9 or 10 students joined. I think 11 was the maximum, I think. So I will see you all hopefully for the next week as well. Okay. Thank you. You guys can now leave the meeting. I will stop the recording here.
Thank you.
Yeah.